this lesson is on DNA replication. If you think about it for a minute, when does DNA copy itself? Before it goes through mitosis or any type of repair, a cell would need to copy all of its genes, so therefore all of its chromosomes, so that the resulting cells have a copy of all the directions on how to make proteins. So I want you to think about this for a minute. Here's my original strand of DNA in red. What's going to happen in DNA replication is these two strands will separate and a new strand will form. This is what we call semi-conservative. One of the strands is new and one of the strands is old. So that is why this is semi-conservative. If it was completely conservative, we would have the red with the red. We would have a complete copy over. But instead, what we've done is we've separated the two strands, used the red as a template for adding new nucleotides in that are complementary. And remember, complementary means the matches. So a purine matches a pyrimidine, A goes with T, and G goes with C. All right, the steps of this process. You are going to want to draw this into your notebook so that you have all the steps. At the bottom of your paper, make sure you have a whole sheet of paper, and at the bottom, make a little bit of DNA and then have it open up wide. I want you to think about a sweatshirt with a zipper, and it's all zipped up, and at the top you unzip it a little bit, and so here's the unzipping, and then here it's all still zipped together. There's an enzyme that does the unzipping of DNA, and it is called helicase. So this enzyme here is helicase, and it is forming what we call our replication fork. So the replication fork is headed in this direction, and it is separating the nucleotides from each other down the middle of the strand. So you could actually see over here A's, C's, T's, G's in a certain specific order. On the other side, you will see their matches. So A would have been with T, C would have been with G, another C, so another G, and T with A, and G with C. We'll say that this side over here is 3' to 5', and we'll say this side over here was our 5 to 3'. The next enzyme that is working is way down here, ahead of the replication fork. So again, if you picture the sweatshirt, it's working down here in the section that has not been unzipped yet. DNA tends to be very tangled and stringy. So the next enzyme is called topoisomerase. Topoisomerase. This actually unravels the DNA so that it makes it easier for helicase to unzip and separate the nucleotides in the double strand. Now, it would not be very easy for replication to occur if there was only helicase and topoisomerase. There has to be something that holds the replication fork open, or these will just hydrogen bond right back together. A G is attracted to a C and forms hydrogen bonds. So we draw in some little dots right here along the side and some over here as well. These are SSBP, single-stranded binding proteins. These binding proteins help hold the replication fork open so that the next enzyme, DNA polymerase, will be able to come in and attach new nucleotides in order. So, in order for this DNA polymerase to work, there has to be a primer. So right here at the top, I'm going to say that this is my primase, and it adds an RNA primer. A primer is a starting point. So a starting point is right here on this 3' side. The enzyme that's actually going to come in, it's attracted to this primer, is called DNA polymerase. So DNA polymerase. The name makes perfect sense. DNA polymer, it's trying to make a long chain of DNA, and ACE, it works as an enzyme. DNA polymerase works off of the template, so this is my template, in the 3' to 5' direction. So on this side, it would add a new T here, and a new G here, and a new G here. And you'll notice that I'm also making the sidebar. The sidebar is the sugar phosphates. It's truly one nucleotide at a time coming in. So phosphate group, sugar with a T. Phosphate group, sugar with a G. Phosphate group, sugar with a G. And it keeps adding them on. My new strand is anti-parallel. So my new strand goes 5' to 3'. It continuously adds because it is in the perfect alignment with the 3' to 5' template being right here. So we call this the leading strand. If you look over here on the other side, it does not work the same way. This side goes 5 to 3, and remember, DNA polymerase wants to work 3 to 5. So what has to happen on this side is the primer is going to attach at some point. If you think about it, unzip a little, primer will attach, work backwards up the strand. Unzip a little further, primer attaches, work up the strand. DNA polymerase can only work in the 3' to 5' direction. So one side is literally perfect. It'll just keep going towards the replication fork in a continuous manner. The other side has to do it section by section. So this is my primer. So DNA polymerase is going to come right in here, and it's going to start adding new nucleotides. It's going to add a G here, and a T here, and a C here, and a C here. Now this little section is what we call an Okazaki fragment. And that fragment is a section of DNA that is attached to the template, and it's building a strand. But it's discontinuous, meaning we have one little piece here. This side just keeps continuing, so as the DNA unravels, it keeps going. What will happen if you think about it, let's say I have a whole bunch more bases here, it has to add a new primer and a new piece. Well, if you put it together piece by piece by piece, this is what we call the lagging strand. So the Okazaki fragments will come together and form what is called the lagging strand. You're probably asking yourself, how does it come together? Because if you have a piece and then another piece, we don't have a full strand of DNA. So we need another enzyme to connect the pieces. This enzyme is called ligase. Ligase is like DNA glue. It connects nucleotides. So ligase will connect the Okazaki fragments until eventually you have two completely new strands like I drew in the first picture. One was formed continuously and quickly. That was my leading strand. One was produced in pieces and then hooked together with ligase. That was my lagging strand. But in the end, I have two strands of DNA. So if you picture for a minute my sweatshirt example, it is all zippered up, and at the beginning, Helicase starts the replication fork and unwinds a little bit of it and opens it up. If this side is 3 to 5, DNA polymerase will attach to that primer and start going in this direction. The other side being 5 to 3, it's going to have to work upwards, and it works away from the replication fork origin.